Hi, Science 11 class. Good morning, good afternoon, or good evening at whatever time you are trying to study this recorded lecture for Module 4 in Science 11. Again, Science 11 deals with contemporary issues in living systems. So from the previous module, in Module 3, you were able to investigate and study cycles in living systems, specifically at the ecosystem level, involving biogeochemical cycle. So the different essential elements and the cycle that is involved with each essential element, you were able to study them. Now we go to Module 4, which is reproductive cycles and patterns. So from the title itself, we will be looking into cycles and patterns in living systems at the reproductive level. So this will be somehow at the cellular level. So you will be encountering a lot of technical terms that pertains to the cellular level of an organism. So as you listen to this PowerPoint, whatever new terms that come to you or whatever new terms that you encounter as you listen to this PowerPoint, you may write them down and then you may always review them to be able to familiarize yourself with those different terms. For our outline in this presentation, it will be into 3 different parts. So the first part of our lecture would be the review of the cell cycle. For the review of the cell cycle, we will go back to our mitosis and meiosis, which you already have discussed during your basic biology, during your high school period, and during your junior or senior high school period. For the second part of the lecture, we will be going into the different reproductive cycles of organisms, specifically haplontic life cycle, diplohaplontic life cycle, and diplontic life cycle. And for the last portion of the lecture, we will be going into patterns in living systems. So these different patterns would be in plant development, pattern formation in animal development, and lastly, the social organizations or social insects. For the last portion, we will be looking into those different or unusual patterns in living organisms. At the end of this presentation, you should be able to first discuss the stages of cell cycle, specifically in mitosis and meiosis. The second one, you should be able to explain the life cycles of selected organisms. And then the third one is to differentiate reproductive processes in flowering plants and humans. And lastly, to appreciate some patterns governing the living systems, specifically the reproductive cycles of organisms. One of the earliest or most ancestral mechanism or way of reproduction are exemplified by bacteria. So a cellular bacteria, having the essential components of a cell, would first duplicate the chromosome. So after the duplication of the chromosome or the genetic material, the cell would now continue to grow. And after cell growth, the bacteria would undergo division into 2 cells. Now we go to cellular reproduction of higher organisms or more complex organisms. This is conducted through the process of what we call as mitosis. So for the process of mitosis or cellular reproduction in higher form organisms, please try to pause from this lecture and try to go or visit this YouTube video for your review. Now, just to give a recap of that video, a cell would have to undergo 2 phases as part of the entire cell cycle. So the first part is the interphase, involving the G1 phase, the synthesis, and the G2 phase. And then lastly, your cell can now enter the mitotic phase or the mitosis. Please remember that your cell cannot undergo the M phase or mitosis without first undergoing your interphase. In your interphase, essential duplication of the genetic material as well as other cellular parts are very important. And then in your M phase or in your mitotic phase, remember again your mnemonics in trying to remember the different stages that happens during the mitotic phase. For the review of your meiosis, please leave first this lecture presentation and then try to visit another YouTube lecture for your review of meiosis. If you have confusions at the first watch of the lecture or the review lecture, you may try to watch it again. Now, just to give some of the most important points in meiosis, remember that meiosis 1, or the first stage of your meiosis, it aims to separate homologous chromosomes. So those homologous chromosomes are first again aligned at your equatorial plate and then they become separated. Now, for the second part of meiosis, or your meiosis 2, it now aims to separate the sister chromatids. To give a summary between your meiosis and your mitosis, let us try to study this Venn diagram. So during your mitosis, the number of chromosomes would remain the same. And then, one cellular division only occurs. One stage of cellular division would only occur from your prophase to telophase. And then the end product would involve two diploid cells. On the other hand, for meiosis, or the cell division that occurs in sex cells, the number of chromosomes are reduced by half, such that the resulting cell would be 4 haploid cells. In this one, you have two stages of cellular divisions that are occurring, which are two rounds of your prophase to metaphase. What's the similarity between meiosis and your mitosis? Both of them would result to the production of new cells, and they both aim to the replication of genetic material. Okay? Again, the major difference, mitosis occurs on somatic cells or on your body cells. Meiosis would occur on your sex cells. And then you also have, again, the specific differences. You may also encounter differences in mitosis between plants and animals. Specifically, you can determine this during the telophase period. So one big difference during the telophase period is that in animals, the cell furrow is being formed. So what happens is that microfilaments form a ring around the cell's equator. And this microfilament ring contracts, trying to pinch the cell's waist. Therefore, it would appear that somehow you have a furrow being formed. And then the waist, quote unquote, is completely pinched off, forming two daughter cells. A different process may be encountered in plants. In plants, instead of a cell furrow, we call it as a cell plate. So a cell plate during the telophase period is being formed in plants. So a carbohydrate-filled vesicles are forming along the cell plate. And then these vesicles would fuse to form new cell wall and plasma membranes between the daughter cells. So you do not observe a furrow that is forming among plant cells. As these vesicles fuse, it is somehow from the inside towards the outside until a complete separation of the daughter cells is accomplished. From that review, I hope that you already gained a mastery between your mitosis and your meiosis. Now, are these processes always accurate? Okay, most of the time, our cells would have this correctional, or they have this quality control points during the cell division process. However, what happens if we encounter a great error in cell division? This may just be one of the results that we encounter when cell division goes wrong. Usually, we encounter cancer cells. So during the normal cell division, the cell division would eventually stop. However, with DNA that would contain mutation, or cells that have damaged DNA, usually cancer cell division occurs where there is non-stop division of your cells. So this cancer, from a long study, have been influenced or can be influenced by a lot of environmental and genetic factors. Okay, so those are just one of the alterations to this pattern in living system, specifically your cell division. For the next part of the presentation, we now go into patterns and cycles, specifically the types of reproductive cycles according to the dominant stage of the life cycle.